All right, we're going to pick up where we left off in class the other night with translation. Now, by now you should have the idea that all of these processes, replication, transcription, and translation, all have the same three stages: initiation, elongation, and termination. And if you remember what initiates each one, the elongation, what components it has, and it's either going to have ATGC or it's going to have AUGC, and then what the termination is. And for translation, it's stop codons. So let's take a look. This is a ribosome, and you know from the last slide that, let me just see if I can show you, that ribosomes in eukaryotes and prokaryotes are quite different. I circled the 16S because that's what we often sequence to find out differences between strains. So let's take a look at this. You should give yourself the quiz.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of three nucleotides on the transfer RNA, called the anticodon, and three nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids.
All right, let's review. The ribosome gets assembled. The mRNA that was produced through what process? Transcription. Loads into the ribosome. These sites are just for reference, but it loads at the P site. A transfer RNA comes in, matches up anticodon to codon, and up here it has the amino acid that matches the anticodon. Remember that T shape with the amino acid? Don't forget about the wobble. This doesn't have to match exactly that third codon. Then what happens next is the next tRNA comes into the A site. Now what is going to happen now? What kind of bond is going to form when these tRNAs move? See that? Right up at the top? That's a peptide bond. So now you're starting to form a polypeptide molecule. Remember that?
As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
How many stop codons are there? 3, right. If this is a prokaryote, the next thing that'll happen is this will spontaneously fold into all the levels of protein folding that you remember. This is the primary structure. The secondary structure is alpha helix or beta pleated sheet, and then there's tertiary and quaternary if necessary. If this is a eukaryote, it's going to go through some processing. We talked about a cap, we talked about a poly A tail, and we talked about splicing if it's a eukaryote. All right, let's go back to our PowerPoint. That's translation. Okay, so you know how we copy DNA, you know how we transcribe the DNA to a message, and now you know how we make the DNA message into an actual protein. How does a bacteria control what proteins get made? And the best example of this is the lac operon in E. coli. And over here I listed your vocabulary: operon, polycistronic, repression, inducible. And This is a really good animation produced by VCell. Basically, an operon is genes all in a row that a bacteria will need to digest lactose in its environment. All right.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Okay. Let's look at this. Go through this. All right, circular DNA, right? Not a plasmid. This is the genomic DNA for the E. coli. This repressor is like a brake. Right now, the enzyme RNA polymerase, which is required for transcription, cannot bind because this is in the way. How do you remove the brake? How do you remove that repressor? Well, you bring in lactose. Once lactose is present in the media, it can bind to, I kind of missed it, can bind to the inducer. I don't want it to start talking again.
Lactose passively enters the E. coli cell and binds to the repressor molecule.
Okay, that's what makes it go away. As soon as the lactose is unavailable, then the repressor is going to bind again like it showed you at the end of the movie. So there's no wasted energy. It only produces those proteins that are required to digest lactose when lactose is in the environment. Now, E. coli prefers to digest glucose, and in your book I've asked a question. Let's go back to the PowerPoint, if you can see it. If you go to the presenter notes, I've asked you what are specific instances where bacteria, your book talks about Pseudomonas, regulates the production of proteins. It gives you 3 times when it regulates the production of proteins. There is a call-out box in your book about Vibrio vulnificus, one of my favorites from this summer. And then I ask you, Why is lactose digestion halted if both glucose and lactose are present? Won't an E. coli break down both of them at the same time? But take a look at that and figure out why. That could be a good question for you. All right, let's continue. So, we've got inducible operons, and they are shut off unless a nutrient molecule is active. There are mechanisms that are the opposite, where a gene is active all the time for a necessary amino acid, in this case tryptophan, and then occasionally, if tryptophan becomes available in the environment, the translation of that gene is shut off. And I'm going to leave you for that to read. Your book goes into it a little bit. I'm not going to hold you responsible for the trip operon, but looking at it may help you understand the difference between something that's turned on when it's needed and then something that's turned off when it's needed. I think it's valuable to look at. All right, now we have to talk about mutations, because now that we make proteins and they work fine, what happens when they don't work so fine and they're not made correctly? There are mutations in the genetic code. Mutations in the genetic code lead to mutations in the mRNA transcript. Mutations in the mRNA transcript lead to mutations in a protein. Structure equals function. So if you don't have the right structure, you're not going to have the correct function. So let's take a look at these. This is my favorite way to look at it. The code is supposed to be 3 letters. That's called a what? A codon. So here are 4 codons, the fat cat sat. And we're going to separate them out into codons. What happens if you get this? What have you lost? And I put this so you could write it in. But what's missing? What changed? You've got a change right here. It's not the anymore, it's this. So what would happen in this instance if you did this kind of mutation? Every single one of these codons, every one of these codons has been changed. So you will no longer get the amino acids that were specified by this. Completely different chain of amino acids. One of these could be a stop codon, so then you won't get the protein at all, or you'll get a significantly different proton. What about this change? This is called a point mutation, by the way. What happened here? Seems we have a little extra, right? This is called an insertion. There's an extra E here, so the first amino acid is correct, but all the rest down the line are incorrect. Now, if you have a mutation that doesn't cause a change in the structure of the protein, all it does is change the wobble, and you still get an amino acid with the same properties, say it's hydrophobic. That's called a silent mutation. It's different if you sequence 2 different organisms, but the protein still works fine because the amino acids maintain their characteristics. A missense mutation means that something has gone wrong and you get a different amino acid, and that can occur through an addition or a deletion, as I showed you here. A nonsense mutation means you end up with a stop codon where you don't want one, and that's what could have happened here. A frame shift mutation is just a way to say that every other codon down the line has been changed. So this is a frame shift. It moved the whole 3 codons down the line. So why don't you take this sentence and see what happens when you remove letters or insert letters and try and see what a frame shift looks like. Those are the kinds of mutations you can have. All right. In laboratory, you used UV rays to try and induce mutations in the DNA of bacteria. Some bacteria, of course, have evolved a way to repair that damage. You can't just go around living outside in the environment and not be able to repair some kinds of UV damage. So some bacteria have this ability, and there are 2 kinds. There are light repair and there are dark repair. So in light repair, it's going to use those photons as an energy source to drive all the enzymes and fix a mutation. In dark repair, it's going to use excision. That means cutting out of the base. That's wrong. It requires DNA polymerase, DNA ligase to put 2 bases back together. Ligase is also used in Okazaki fragments. Keep relating your information back to things you already learned. Photolysase is going to be an important enzyme, and methylated DNA we're going to run into. If you remember from chemistry, a methyl group is a carbon and 3 hydrogens, a CH3. and they're going to be very important in how a bacteria can tell which strand to fix. Thymidine is a base. A dimer means there's two of these, and they're going to become connected together through the action of UV rays.
When cells are exposed to sunlight, radiant energy can damage the DNA. For example, ultraviolet irradiation causes covalent bond formation between adjacent thymines on the same strand of DNA. When DNA is damaged in this way, it cannot be replicated or transcribed. Most cells, however, are able to repair damage to DNA. For example, an enzyme called photolyase can break the covalent bond between the thymine dimers. This enzyme requires visible light, which is used as the energy source for bond cleavage. Hence, this mechanism is referred to as light repair. A second repair mechanism involves an enzyme that excises the damaged segment from a single strand of DNA. This process is termed excision repair or dark repair and does not require visible light. The excised nucleotides are then replaced by DNA polymerase. DNA ligase forms the final phosphodiester bond.
Isn't that nifty? That is just great. They've got an enzyme powered by light. And if you don't have any light available, you can use excision repair. So what would happen if you had taken a strain of bacteria the other week in lab, you had subjected it to UV rays, and you kept it in an incubator? Think about that. That's a good question. What would happen? What repair mechanism would it need to use? Would phytolysase be available? All right. All right. I want you to notice down in the notes that I've got questions for you. Repaired by, define mutant. There's some nice questions there. Okay. The Ames test. What is the aim of the Ames test? That's just silly. I actually put a Wikipedia thing here for you to read about the Ames test. It's a pretty good description, and it doesn't go crazy, and it's one of the most important tests that we have for new chemicals entering the market. So basically you take rat liver, and we're going to come back to liver extract, and we're going to take a Salmonella strain that is a mutant. It's a histidine mutant. It requires histidine, the amino acid histidine, in its media to grow. So this means if you have a plate and you put this mutant on it with very little histidine, it's not really going to grow. Remember that lag phase? It's not really going to grow because there's not enough histamine for it. So if you then put a chemical on it that is mutagenic or carcinogenic, in other words, it's capable of changing the DNA of something, and then all of a sudden these colonies start to grow like crazy, you know it's a mutagenic chemical because it induced a mutation in the Salmonella so that now it doesn't require histidine anymore. This is a relatively easy mutation to achieve if a chemical is mutagenic. All right. In the control plate, you would only have 1 or 2 colonies that would spontaneously revert. So you can tell the difference between 2 plates. If there's a lot of colonies, it was a mutagenic compound, and this is a plate you did nothing to, and only a couple colonies had reverted. So the Ames test is very important for deciding whether right off the bat a new chemical is mutagenic or not. It does not stop a chemical from coming to market, because they will go on and test it in mice and other model organisms. But I said we would come back to liver. So what happens when a chemical hits your body is that your liver has to process it. Your liver detoxifies everything. So if you put liver extract in here, it's possible that the compound might not be mutagenic because of the way the liver breaks it down, and therefore you get the same kind of plate. It is also possible that the compound is not in and of itself mutagenic, but once the liver enzymes process it, some of the breakdown products of it are mutagenic, and then you would get this kind of plate. Think about alcohol. It's not necessarily bad for you as alcohol, but you get a hangover because when it hits your liver, the enzymatic breakdown products of alcohol make you miserable, acetaldehyde. So that's why liver enzymes are included in the Ames test. All right, this is 3 ways that bacteria can gain new genetic traits. 3 ways. I call it sex, drugs, rock and roll. It's much funnier in person, but the first way is conjugation. This is the most popular way that bacteria gain antibiotic resistance plasmids. Basically, it's the sharing of plasmids across the bacterial world. So here's our donor, and here's an F plasmid, fertility, a conjugation plasmid. This plasmid allows a bacteria to grow a conjugation pilus. Here's a bacteria just minding its own business, doesn't have a fertility plasmid, comes up against one of these bacteria that does. Bing, the pilus meets the other bacteria. This plasmid is replicated, and as it's replicated from the point of origin, one strand of DNA goes over to the other cell. This double strand is recompleted because it's complementary base pairing, and then in the recipient cell, that single DNA strand is made complementary, and now it too can produce a pilus. And if this plasmid had antibiotic resistance on it, say resistance to kandomycin or ampicillin or vancomycin, this bacteria has now become resistant to the antibiotic. This is very, very common, and antibiotic resistance spreads incredibly quickly in that fashion. And I've given you one of those nice little animations for you to look at. Here's a vocabulary associated with conjugation: F plasmid, F plus, a conjugation pilus. And a high frequency of recombination cell is a special instance where, you can watch it in the movie, but it's a special instance where this plasmid isn't the only thing to go to the recipient cell. There can be special instances where another trait allows the genomic DNA to be copied as well. Now it takes about 100 minutes for E. coli to be in contact with another cell for its entire genomic sequence to be run off into a recipient cell, so it's incredibly rare for an E. coli in your intestines or in the environment to be in contact with another bacteria for 100 minutes. But it can happen. You should know that high frequency of recombination means that a cell is capable of running off its genomic DNA as well as its plasmid DNA. All right, transformation. This is a special instance where naked DNA from a dead bacteria can be taken up by a live bacteria and receive new traits in that way. And the person who told us about this was Frederick Griffith before he died in World War II, and then Avery, McCarty, and McLeod, and I hope you've heard of those guys in biology, figured out this transforming principle that naked DNA can be taken up by a bacterial cell from the environment, integrated into its own DNA, and therefore confer new traits. And the experiment was a rough strain of pneumonia, which did not cause disease, a smooth strain, which did cause disease, virulence. It was called smooth because it had a glycocalyx layer, a capsule. And if you remember from the chapter on structure, a capsule can make a bacteria more virulent because it sticks more easily to us. What Frederick Griffith did was to heat kill the virulent strain of pneumonia. So it was a lot of dead bacteria. And he knew it was dead because when he injected it into a mouse, the mouse lived. So the mouse lived after being infected with a smooth, heat-killed strain, dead bacteria. And then for some reason, he decided to take that heat-killed bacteria and mix it with the rough, the bacteria that does not normally cause disease. And what happened is, see, it didn't cause disease, happy mouse, happy mouse. Now you've got heat-killed and a bacteria that does not cause disease, and you've got a dead mouse. And he died before he could figure that out, but he called it a transforming principle. Avery, McCarty, and McLeod came along and through various enzyme analyses discovered that the only agent capable of doing that was DNA. That is one of the foundational experiments for demonstrating that DNA was the hereditary material. So transformation, uptake of naked DNA from the environment to confer new traits on an organism. And when we use antibiotics to kill bacteria, if we kill bacteria that have F plus plasmids, can that DNA be taken up through transformation? You betcha. All right. Transduction. Transduction involves a virus. We need a virus as a vector. A vector is a third organism that acts as a transfer device between two other organisms. So in this case it's viruses. No, they're not alive, but they cause a lot of trouble because they bring their own genetic material into bacteria and into us. It comes in two flavors called specialized transduction and generalized transduction. And I think the important thing to remember about this is that in generalized transduction, you have a virus cause the complete destruction of a cell. All of the DNA ends up completely randomized, both the bacteria and the viral DNA. It gets packaged into new viruses, and every once in a while, some of the DNA from the old bacteria gets packaged into a virus head. And then that can go on to infect a new cell, and that DNA from one bacteria gets shuttled via a virus into a second bacteria. Now the virus is no good, but now that bacteria's got a new trait. So that's generalized. Every once in a while, some bacterial DNA gets packaged in a virus which infects a cell, and now that cell has a new trait. So this is one way that viruses can confer and transfer new traits. I think of it like viruses are a cargo carrier. In specialized transduction, this is when each and every virus will carry a particle of the bacteria. So right here you have a virus integrating itself into the host DNA, and then when it takes itself out again, it takes a piece of the bacterial genomic chromosome with it. And then that ends up in each and every instance of the virus, and every time that virus goes on to infect a new cell, it takes a new trait with it. And you have a video here. I would do the quizzes on transduction. Students seem to have a lot of trouble with transduction. That's it. That's the end. So I'm going to leave this up to you because we have to continue on with class. We can't get too far out of sync with the other class. Email me questions, see me in lab. I'll be happy to answer questions. I sat with a couple students the other day and they had great questions. All right, I will decide by Monday if we're going to still have our genetics quiz on Wednesday. Thanks.